Now, let's discuss the implications of a bactericidal agent. What happens when the agent is bactericidal? Cidal is killing effect. So because the organism is killed, the infection is totally eradicated. And therefore, there is less chances of relapse. There is less chance that the organism will exist in a dormant stage and will multiply later. So, total eradication and less chances of relapse. Example, there is a disease called enteric fever or typhoid fever, for which initially in the olden days we used to use chloramphenicol, which is a bacteriostatic agent. And with this agent, we used to have a lot of relapses. That was the issue. When the newer drugs came, we got cotrimoxazole, ampicillin and amoxicillin, ciprofloxacin, and now the latest one is ceftriaxone. All of them are cidal, so the chances of relapse in enteric fever are almost over because we got cidal drugs now. Another thing is when the drug is cidal, there is a rapid killing effect and this shortens the length of treatment. We have a very classic example of tuberculosis. In the olden days, in 50s and 60s and 70s, tuberculosis was considered as a very severe illness because it needed treatment for a pretty long period of time because we didn't have effective drugs. As we went on having effective drugs, like the cidal drugs in the form of streptomycin, INH, rifampin, pyrazinamide, the length of treatment for tuberculosis has remarkably become less. Now let's discuss the combinations. There's a very remarkable combination called a combination of sulfamethoxazole and trimethoprim. Both these drugs are static, but when you use them in a particular ratio, the combination becomes cidal. Because the two drugs, sulfa and trimethoprim, they act on two consecutive steps in the folic acid metabolism. This is called sequential blockade, and this leads to synergism. What it means is two static drugs, when used individually, they are bacteriostatic, but when you use them in a combination, they become cidal. The second purpose of using combination could be to cover a broader spectrum of activity. And another third purpose, other types of combination. For example, penicillin is combined with clavulanic acid. What is clavulanic acid? Clavulanic acid is a beta-lactamase inhibitor. Penicillin is likely to get destroyed by the organisms which produce beta-lactamase enzyme, and clavulanic acid destroys the penicillinase enzyme or the beta-lactamase enzyme produced by the bacteria. So clavulanic acid is going to protect the penicillin and is going to help the action of penicillin. So this is another different type of combination which could be used in practice. Now let's have a list of some static and cidal drugs on this particular slide. Let's have a look at the slide. So you have sulfonamides and trimethoprim. Just now we said they are bacteriostatic. They are coming on the left side, static. And if you go to the right side, down here, I have shown cotrimoxazole. So that's the combination of sulfonamide and trimethoprim. On the left side, static, there is erythromycin. The next two agents on the left side, static, are tetracyclines and chloramphenicol. And these are broad spectrum agents. So I think this would be worthwhile to remember the two agents which are broad spectrum. We call them broad spectrum, but they are bacteriostatic. They are not cidal. After this, I am mentioning two anti-tuberculosis agents, thiacetazone and ethambutol. They are also bacteriostatic. The next agent is dapsone, is an anti-leprosy agent, also has got predominantly bacteriostatic effect. And the last one in the left column is pyrimethamine. This pyrimethamine also is a bacteriostatic agent. Of course, this is an anti-malarial drug and this could be used in the management of malaria. Let's come to the right side. Right side is the bactericidal drugs. Amongst the bactericidal drugs, we have important drugs. Those are the beta-lactam antibiotics, that's penicillins and cephalosporins. Penicillins include penicillin G, ampicillin, amoxicillin, carbapenicillin. All of them are penicillins, and they are the bactericidal agents. The second group of beta-lactam antibiotics is cephalosporins. Because they are beta-lactam, they are also bactericidal. Both these penicillins and cephalosporins mainly act on the cell wall synthesis. Next we have aminoglycosides. Look at the slide. Streptomycin, gentamicin, amikacin, tobramycin. All of them are aminoglycosides. They mainly act on gram-negative aerobic organisms and they are bactericidal. Going down on the right side, the next group is quinolones and the fluoroquinolones, especially, for example, ciprofloxacin. What I have shown here is, I have avoided the word floxacin and I have just written the suffix. Cipro, O, Pe, Lome, Spar. That's ciprofloxacin, ofloxacin, pefloxacin, lomefloxacin and sparfloxacin. All of them are fluoroquinolones and they are bactericidal in nature. Down we mentioned two more drugs, that's metronidazole and tinidazole. These are used mainly for amoebiasis and they are also used for gram-negative anaerobic infections, and metronidazole and tinidazole are the bactericidal agents. Then of course there is cotrimoxazole, about which we already said that it's cidal, and the last line is about the three antituberculosis agents. INH, that's isoniazid, then there's rifampin and pyrazinamide. All three are bactericidal agents. Along with these three, there's one more agent above under aminoglycosides, that's streptomycin. So streptomycin, INH, rifampin, pyrazinamide, all four bactericidal antituberculosis agents who have produced a revolution in the management of tuberculosis. Let's have a look at the slide again and review it, how to remember static drugs and cidal drugs. Drugs which inhibit cell wall synthesis are going to be cidal, and they're beta-lactam drugs, penicillins and cephalosporins. Drugs which inhibit the gram-negative organisms, aerobic as well as anaerobic. Aerobic is aminoglycoside and anaerobic is metronidazole, tinidazole. They are going to be bactericidal. Drugs which inhibit gram-negative organisms, one more group, fluoroquinolones, are bactericidal. So that will help us to remember what are the bactericidal agents. Coming to the left side, that's the bacteriostatic drugs. You have sulfa and trimethoprim on the left side because they are static, and the combination is mentioned on the right side, that's cotrimoxazole, that's cidal. Next, erythromycin is static, and the two broad spectrum drugs, tetracycline and chloramphenicol, they are also static. The remaining left side comprises of pyrimethamine, antimalarial drug, static, and two agents anti-tuberculosis, that's thiacetazone, ethambutol, static, and dapsone, anti-leprosy drug, bacteriostatic. So I think this slide is going to be helpful to you to remember which drugs are primarily bacteriostatic or primarily bactericidal. Next we go on to understand what's the mechanism of action. This topic is totally different from the rest of the chapters in pharmacology, because when you discuss about the pharmacotherapy of various illnesses, you think of a drug going in human body and acting on the human cells and producing some interaction with the human receptors. Now this is something different. You are giving a drug inside human body, but you don't expect the drug to act on human body. You expect the drug to act on something which is inside you, and that's the microorganism. And you expect the drug to selectively act on the microorganism and do not start destroying the normal human cells. So it's essential to know what's the mechanism of action of various antimicrobial agents. We'll do it in the form of tables, and we'll go to each part of the cell and we'll know which drug acts on this part of the cell. Number one, we consider the periphery of the cell. That's the cell wall and cell membrane. Of course, we're talking about the bacterium. Drugs which inhibit cell wall synthesis are the beta-lactam antibiotics, penicillins, cephalosporins, other beta-lactam drugs like imipenem and meropenem and aztreonam. Then you have an important drug in this group, that's vancomycin, also cycloserine and bacitracin. And just to complete the list, I have added one agent from tuberculosis, which inhibits cell wall synthesis, that's your INH. I hope you're looking at the slide so that you can realize what I'm saying. The second group on this particular slide is the drug which damages the cytoplasmic membrane. So it's also like inhibiting the cell wall synthesis or acting in the periphery and not acting in the center. Drugs which damage the cytoplasmic membrane, they include polymyxins, colistin, and polyene antibiotics. So this slide tells you about the drugs acting on cell wall and cell membrane. We move on to the next slide, drugs which inhibit the protein synthesis. And in the left column, I have differentiated as 30S and 50S. What is this 30S? Drugs inhibiting protein synthesis by binding with the 30S ribosomal subunit or by binding with the 50S ribosomal subunit. Binding with 30S ribosomal subunit is aminoglycosides. What do? They inhibit the peptide formation, they inhibit the formation of initiation complexes and produce misreading and produce polysome breakage. Whereas drugs which combine with the 50S ribosomal subunit and inhibit protein synthesis are macrolides. That's erythromycin, clarithromycin, etc. They block the amino acid translocation reactions and formation of the initiation complexes. So two drugs on this slide inhibiting protein synthesis by combining with 30S or by combining with 50S. There are more drugs which can also do it by combining with 30S and 50S, which we'll see on the next slide. Inhibiting protein synthesis once again, but I have put them separate because they are broad spectrum static drugs, that's tetracyclines and chloramphenicol. The tetracyclines combine with the 30S ribosomal subunit and chloramphenicol combines with the 50S ribosomal subunit. Tetracyclines prevent the binding of amino acid tRNA to the acceptor site on the mRNA ribosome complex. I hope you are understanding. They prevent the binding of amino acid tRNA to the acceptor site. Whereas chloramphenicol inhibit peptidyl transferase. It inhibits the peptidyl transferase. So this is how the two broad spectrum antibiotics, they also inhibit the protein synthesis. We move on to the next way or next mechanism of action through the next slide. There are drugs which inhibit nucleic acid synthesis. A very typical example is rifampin. It is an anti-tuberculosis agent and it inhibits the enzyme DNA dependent RNA polymerase. I hope you learnt it in your undergraduate days, and it's essential to remember the name of the enzyme for rifampin, DNA dependent RNA polymerase. The next mechanism inhibiting DNA synthesis is metronidazole tinidazole. The next drug on the slide is quinolones. Quinolones also have a specific enzyme for action. They inhibit the enzyme DNA gyrase and inhibit the nucleic acid synthesis. You can also call this enzyme topoisomerase. Finally, we need to understand that the microorganisms needs certain metabolites, and these metabolites inside the microorganism is going to help them to do their DNA synthesis and multiplication. So if we could use a drug which could be anti-metabolite or make the microorganism devoid of the important metabolite which they want, then we could inhibit the microorganisms. So the next row is speaking about anti-metabolites and the drugs which are folate antagonist. There are many drugs which are folate antagonist. It includes sulfur drugs, dapsone, trimethoprim. So the sulfur drugs, dapsone, trimethoprim, all three are static drugs. Now if you combine sulfur with trimethoprim, you get cotriamoxazole, which is a cidal drug, and finally you have pyrimethamine. That's an anti-malarial drug. Many anti-malarial drugs are anti-metabolites or they are folate antagonist. So that's about a novel mechanism of producing anti-metabolite action. Then let's try to know what is spectrum of activity. Spectrum is the range of microorganisms which are inhibited by a drug. If a drug is able to inhibit a small number of small group of microorganisms, you call it narrow spectrum. For example, penicillin G or benzyl penicillin, the first penicillin, mainly inhibited gram-positive organisms. Or the aminoglycosides or metronidazole mainly inhibit gram-negative organisms. The next concept is broad-spectrum antimicrobial or broad-spectrum antibiotic. BSA, broad-spectrum antimicrobial. And according to classical pharmacology, there are only two agents which are called broad-spectrum agents, and that's tetracyclines and chloramphenicol. I hope you realize this is a very rigid classification, and there could be someone who has got little broader spectrum than narrow spectrum, or who has got little narrower spectrum than broad spectrum. I hope you are listening to me. I'm taking you to the next slide. Have a look at the slide. I've written it here. Broader than narrow spectrum or narrower than broad spectrum. So you can't put it in narrow, you can't put it in broad. So we have a beautiful term for this. It's called extended spectrum antimicrobial agents. For example, we have penicillins. You can see on the slide in blues, I have indicated the extended spectrum drugs, extended spectrum penicillins. There are many penicillins who also act on the gram-negative organisms. So we should not call them broad spectrum antibiotics. We call them extended spectrum penicillins. Then you have second and third generation cephalosporins which act against many gram-negative organisms. So cephalosporins, second and third generation. Then we have cotrimoxazole, which has a broadened spectrum of activity. We put it under extended spectrum. The next one is sulfur drugs and trimethoprim. Do inhibit many gram-positive, many gram-negative, many atypical organisms. Then we have quinolones. They act mainly on the gram-negative organisms, but do have an extended spectrum. So all these drugs we will call henceforth as extended spectrum antimicrobial agents. And if you classically want to talk about broad spectrum, it will be only tetracyclines and chloramphenicol. Finally, on this slide you see macrolides have also got extended spectrum because they act on many atypical organisms like mycoplasma. So that's the discussion of the spectrum of activity. Now we come to the next slide with the discussion of bacterial resistance. And sometimes I wonder, it's difficult to decide who is wiser. Human being is wiser or the microorganism is wiser. The microorganism prevailed much before us on this earth. And I feel microorganisms are much wiser than the human beings. You designed the drugs to kill the microorganisms. The microorganisms found out what's the mechanism of action of your drug, and the microorganism tried to overcome the mechanism of your antimicrobial agent, and they practice some other mechanism so that they can withstand or they can be resistant to your drugs. When this happened, the human beings also tried to overcome the mechanism by the microorganism. This particular battle is on and on for years. So we talk about bacterial resistance. Bacterial resistance becomes a problem especially in the post-operative infections or hospital infections where there could be a lot of microorganisms and they could become resistant to the antimicrobial agents. Three important organisms to be remembered are Proteus, Klebsiella and Pseudomonas. They are very vulnerable for bacterial resistance. The next important organism which has troubled mankind since many years is the Staphylococcus. And the Staphylococci have become resistant to many penicillins. Then you started calling them methicillin resistant Staphylococcus aureus. Look at the slide, MRSA. Then we had vancomycin to fight with these organisms, but organisms also started developing resistance to vancomycin. So it's called vancomycin resistant Staphylococcus aureus. That's VRSA. Next important organism is Enterococcus. That's Streptococcus faecalis and Streptococcus pisium. These are difficult organisms to eradicate. Next, you have Salmonella typhi, organism producing typhoid fever or enteric fever, also started developing resistance to the routine antimicrobial agents. For example, it became resistant to chloramphenicol, it became resistant to ampicillin, amoxicillin, it became resistant to cotrimoxazole. Then we also now have ciprofloxacin resistant strains. And now we have the final answer, that's ceftriaxone, to fight against Salmonella typhi. The next organism which can develop resistance is Neisseria gonorrhoeae, and a lot of Neisseria gonorrhoeae organisms have started developing resistance to penicillins. Finally, I am mentioning Mycobacterium tuberculosis. Tuberculosis once upon a time was a very difficult disease to eradicate. After we got many antimicrobial agents, we thought we have conquered tuberculosis and the incidence of tuberculosis would fall now. Fortunately, the picture was good, and later came an immunodeficiency syndrome called HIV disease. And due to the HIV disease, Mycobacterium tuberculosis started developing again, and now we have many cases of drug-resistant tuberculosis. So even as of now, tuberculosis remains in discussion, and there are resistant Mycobacterium tuberculosis organisms. So this was an overview of the resistant organisms. What can the microorganisms do? See, it's very simple. If you want to fight a drug and you want to become more resistant to the drug, you want to resist the action of a drug, just go logically, think logically, and you can get many mechanisms. Look at this slide. Number one, you have given a drug to the microorganism to kill it. I am a microorganism. What I can do is, I can synthesize enzymes to destroy your drug, to inactivate your drug. So synthesis of enzymes to inactivate the drug. That's the first simple mechanism. The second one which I could do as a microorganism is to find out where is your drug coming and binding to me. Find out where is it going to bind, and then I'll do something, some change in my structure so that I change my binding site so that your drug cannot bind at all. That's the second way. So to prevent or inhibit the binding of the drug. The third simplest way the microorganism practices is when your drug is coming inside, either it will not allow the entry of the drug inside, or after the drug comes inside, it will throw it out. This is called a transport mechanism or efflux. Efflux is throwing it out. So that's the third common mechanism the microorganism can practice. And next, the microorganism can synthesize substrates to overcome the inhibition of metabolites. As we saw, there are anti-metabolite drugs which are going to produce anti-metabolite effect in the microorganism. So if I am a microorganism, I'll synthesize the substrate in a very large amount so that your drug cannot inhibit or cannot overcome the substrate. So these are four basic mechanisms which are practiced by the antimicrobial agents by the microorganisms, to produce bacterial resistance. So antimicrobial agents and the bacterial resistance is an important issue.